Okay. Okay, everyone. Thank you all for joining us. Welcome. Appreciate your patience while we get started. Today, I need to remind you, as usual, that we are recording this and live streaming it on YouTube, and that the recorded conversations will be made publicly available. And so if you don't want to be seen or heard, please make sure that you don't speak or include yourself in the meeting. It's just a legal formality that I do have to say that. I want to thank you again. This is our last meeting in February. As usual, I need to remind you that the leaders of the group are myself and Reinhard Labenbacher, with the able assistance that you from Jim Sluka and Bruce Shapiro. Reinhard and Bruce are not available today. It's study section time, so they're off on the relevant study section. As always, I'll also remind you that we have a Slack channel. We really would appreciate it if people would use the Slack channel. There's a transition to more Slack usage for the whole IMAG MSM project. And if you have any issues with that, please contact us. As always, I also need to tell you that we'd appreciate your updates and improvements to the IMAG wiki. I realize that editing wikis is not the most exciting thing to do. It's time consuming. Content generation is always a bit of a difficulty, but the more people use that, the more useful it will be. Rules of the meeting. This is a very collegial meeting. We always appreciate questions. We'd especially appreciate questions from people who don't usually speak up. And if possible, we'd like to have focus on more general questions. I didn't clear for this week whether Jim Fetter and Rita would be available after the meeting to talk to people in more detail. If you would like that today, I can set up a breakout session for that, but it wasn't set up for this week. We're going to start that next week, more formal.
Jim did ask me about that, and I said it was okay.
Oh, it is? Okay. And Rita?
It's okay. I said yes.
Can you
okay.
stay on for next? Okay. So I will.
Yes.
So then I will leave the meeting open. I don't have the breakout room set up right now, so it may take a little bit of doing to get the breakout set up appropriately, but we'll be able to have a little bit more informal discussion for 30 minutes after the close of the formal meeting. So thank you both to both of our speakers for being willing to do that. And then I'll give people a 5-minute warning, as usual, during the presentations. For the upcoming meetings, we have Yi Zhang from Georgia State speaking next week, and Ellen Kuhl. The following week, we have Robert Stratford, who'll be talking about drug development issues, and Veronica Zanitsina from Emory talking about the subgroup plan for innate and adaptive immune system response. We'd like very much if in the coming weeks, the subgroup leads would be willing to present where their subgroups are. And so we'll try to block out the mini seminar slots to allow subgroup leaders to present and have a bit more active discussion in the main meeting. And so if you are the leader of a subgroup who would like to present, please let me know, Reinhardt know, and Bruce and Jim Sluka know, so that we can get you on the schedule. And we'd always like to have your volunteer for speaking in the mini seminars. We'd love to have your suggestions for people to invite. We're going to have Denise Kirshner coming up, and a few other people that are very heavily involved in this kind of work, and that will be quite interesting. So without more ado, I'd like to turn over the first half of the mini seminars to Professor Rita de Almeida from the Universidade Federal do Rio Grande do Sul in Brazil, in Porto Alegre, Brazil. and she's going to be talking to us about transcriptogram analyses for SARS-CoV1 and CoV2 immune response. Rita, I'm
Thank you,
going to
James.
turn it over to you.
Okay, I will. I have to share my screen, right? Okay. So this is, first of all, I'd like to thank you and the organizers for the opportunity to speak about the work that we have been doing. It's a collaboration between me and Gilberto Thomas, who's also a professor at Porto Alegre, and James Glacier. Okay. So we have applied a technique of bioinformatic analysis that we have been developing in Brazil for, I don't know, over a decade. So let's go to the, I don't know if I had the slide transition. Okay. So there, I don't know why it's not. Okay, so just have to, okay. So the viral pandemia, it requires effective therapy to treat for the patients, okay? First of all, we have the strong immune responses are essential to contain and clear the viral infection. But then, on the other hand, you have excessive inflammation may damage tissues, delay tissue healing after viral clearance, and lead to acute inflammatory responses or sepsis. In the case of SARS-CoV-2, the severity of immune response pathologies differ greatly between individuals. And with the current knowledge about the disease, we cannot predict the response in an individual such that we could advance the therapy before something bad happens. Okay, so we don't have this knowledge. So the individual therapy requires that we associate information on the patient immune response evolution with the clinical symptoms and the infection spread and intensity. Okay. We need, for this modeling, for this, to make this model, to make these ideas, we need quantitative models. Okay, the modeling of the immune response to SARS-CoV-2 critically depends on the identification of the most important biological mechanisms which act at different phase infection. So what are the differences, molecular differences in the immune response in different patients? Why do they, react differently? The point is the immune system is very complex. And so if you have to answer these questions in a timing that is effective for therapy, we need to design personalized therapies, and we need to quantitative models. Okay, to construct such models, we need to associate information on the patient response and what's happening inside the patient, and we need what's happening to the viral load and also to the symptoms. Okay, it's going on, it's. Okay. So what kind? Okay, I understand what I'm doing. Sorry. So what kind of data do we need? Okay. Well, we need the evolution, the time evolution, because what's happening, it happens in a very fast way inside them. You know, the viral enters and sometimes the patient is well, and then in 2-3 hours it gets really worse, and you didn't know if you could expect that or not. Okay, so maybe the information of these outcomes or these reactions are in some measurements that you can do about how the expression of some genes or the protein profile or some other places in that tissue or in another tissue. And so we have to integrate the extensive data from in vitro, organoid, and animal experiments with the more limited clinical observations in humans. And when you have, of course, you have to have these measurements of transcriptomes and the protein profiles. And when doing that, I mean, everybody, I mean, not everybody, but some of you are familiar with transcriptome and protein profiling. And the point is the statistical analysis is critical. And the point is, these transcriptomes should search for the sets of covariant genes analysis. You should have some analysis to interpret, and you should look for them. Okay, so so the idea is time and patient matched data on viral load and associated with protein profiles and transcriptome from different tissues and organs. Of course, it's not always possible, right? Because you cannot do that for humans and people who are diseased, and well, there are limitations on that. But you can do for different tissues, and you can do for different cell lines, and you can do maybe with some animal models. Okay, one part of the required measurements are transcriptomes and protein profile. And this is the focus of my talk. First of all, I'm going to talk about transcriptomes. The example here are for transcriptomes, not for protein profile. But they are, I mean, it's to adapt for protein profiling is direct, okay? So first of all, you have the transcriptomes are produced by either RNA-seq or microarrays, and the profile of the RNA messengers are, that's what they do. I mean, they do the profile of the RNA message, mRNA, produced by the cells. The human cells has of the order of 30,000, and many of these genes are not well characterized, about 2/3, depending on the definition of how well characterized. Another problem with these measurements is that the noise is a problem because you have biological variance and technical variance, and then you can have difficulty in detect the changes in gene expression due to a treatment or a different condition. This is, I mean, this is well known that these measurements have these problems. Okay, so I'm going to do what Transcriptograms, which is this bioinformatic tool that we developed, what it does to deal with the noise that is typical of these experiments. For example, in this first panel here, okay, which is set random and R equals 0, what we have done is just we collected, in a way that I am going to explain in a minute, 10,000 genes of the human genome. And then for one given experiment, we just compared or list the full change of each one of these almost 10,000 genes that are here. They are ordered, these genes, they are randomly ordered. But I tell you, the way we have selected them is focused on how important or how connected to the other genes they are. So we have some scheme to order these genes. And to order the genes, I mean to cluster the genes on this line by the way they interact or the way they are associated. If they participate in the same biological function or in the same KEGG pathway, okay? And so there is, well, you can see there are some differences. There are more altered regions than others, but again, it's very difficult to say that there is. Now, what we do here is to transcriptograms. It's for the same data, but we do a kind of a window average over this ordered list. Here is random list, this is orderly. And then this window average, which comprises about 60 genes, what they do is that they do noise, which is random, okay? And by doing this, it really decreases the standard deviation of the number you assign for each one of the genes. And here is, and in the line equal 1, we have the divided or by the control, which is equivalent to do something like a fold change, right? So in here, you see this small gray area, which is the standard error for the control sample, and following the red, which is the, you know, the sample that you are interested in calculating, is it shows you some differences, and it's followed by red shadow as well. Well, the pink shadow. The shadow. What this pink is doing is representing the standard error. What you see here, now you here and you look here. The difference is that here the signal is much more clear, and you can see what are the regions of this ordering thing that is altered with much better precision. Okay, so the point is, if you see some experiment and make a transcriptogram and you see a picture like this, what does it mean? I mean, what are these genes that seems to be so more intensely expressed than the control? Then you have to know what are the genes here, but you use always the same ordering. So here is 0 to 1 because we divided by the number of genes that we have selected, and because sometimes we want to do some evolution analysis, so we compare different organisms. But here, what you see is that we have projected some gene ontology terms or KEGG pathways. For example, in this big peak here, which is associated to mRNA processing, what we did is just to tell you what is the probability that in this region you will find a gene that belongs to this term. Okay, so you see something very, very small, and then it jumps almost to 2.9. So a window of 60 genes, 30 genes to one side, 30 genes to the other side. What you see is that the probability is almost 90% are belong to the same thing. And then you do it for all the ordering, and then you project different terms. What you see here is that you have terms that have to do either with RNA processing or with DNA repair, you have a region that is associated to cell cycle, okay? And then here you have signaling like Wnt, like G protein signaling, and then you have things like regulation of small GTPases, lymphocytes, and then you have, for example, here you have things that has to do with the ECM, and then here you have something which is typical of energy metabolism. You should expect this thing. I tell you, what happens is that when we repeat it, I mean, we didn't produce artificially these things. It was not curated to obtain disorder. I will explain in 1 minute. And what happens is that when you do the same process for other
Just
organisms,
a 5-minute warning.
okay. Oh my God. Okay, so we have done it for some SARS-CoV1, and we have done the transcriptograms. Okay, this way of analyzing. It was node data. It's from 2013, and they consider CALU3 cell line selected for expression for ACE2 at MOI 5. And what is wonderful, they recorded it for all these time steps, okay? And they have microarrays, robust microarray analyzed. And then, oh, I'm sorry, okay. So what we hear, you have these mock infected samples of this CALU3. They have this mock, and they have this infected with SARS-CoV1 virus. So in the beginning, when they have 0 hour after post-infection, what you have is something like that. So you didn't see any alteration whatsoever, nor for mock, not for WT, for the wild type. But after 24 hours, both mock and wild type present very impressive, very different. I mean, they present some alterations that are significant, and you see that in some parts they are different, but in some others they are not. After 48 hours, the mock, the wild infected, what you see is that it's really more, it's completely different from the blue, but again, the blue have some very important things. So what we did next was to use as control, not the mock 0, but use to, to analyze the wild type relative to the same time mock samples. So what you have here is the difference between infecting with a virus or not infecting with a virus. And you can see that in the beginning, they are like that, okay? They are not almost no difference, but after some time, 24 hours or 48 hours, you can see some very impressive peaks of and depressions that has to do with the differential expression of this thing. Okay. So here you see what happens as time goes by. Okay. So you have here the time is passing, and that's what happens to the transcriptogram or to the profile of the thing. Okay. So what we have to do is just to look at these things and see what are these peaks, which are important alterations that we have. So we chose the peaks, okay? And, sorry, we chose the peaks using some criterion. And for each one of the peaks, we saw what happened to that peak and analyzed the genes that were twice, but genes without transcriptograms now, and that they were presenting fold changes that are larger than 2, either for more or for less. And we did it for all the peaks that were significant, and we could find 219 genes, okay? So we just observed that some of the genes were covariant, they had the same. So what we did was to consider the covariance matrix for all these genes, and we could cluster them in 3 different clusters, cluster A, cluster B, and cluster C, which have different behavior, okay? So here is the covariance matrix. This is for this cluster here, which is cluster A, which goes from 1 and then goes back, goes down. And then you have this one, goes up and then goes down. This line here is the average of them. And then you have the same average here. And these guys go, it's different the way they go. Okay, here is, you can have something which you can see all the congregation of these things. And you see that, and in black, what you see is the viral load. Of course, they are in different unities here and here, but you can see what's happening. And you see that the blue, the B cluster, almost follows the viral load. And while the green one is just goes, you know, it goes up, it doesn't go down just like that. And this one is just the mirror of the green blue. These guys here are enriched with the mitochondrial things. And these guys are essentially the immune response. So here I have to put it here.
If you could try to wrap up in another minute or two, please.
Okay. So what we could do and see that the blue thing is doing is playing the role of anti-inflammatory thing. And this is only inflammatory. You can see that because we look to the HSD1B, which are in fact the cortisol. And one of them is anti-inflammatory, the B2, and B1 is inflammatory. And what you can see is that something that increases inflammation is in gene green and cluster green, and this is in the cluster. Because of that, we could see the difference and the difference of these two genes. These two clusters, the genes in two clusters, are spread in the ordering, and what's interesting is that you have to pick them up. But this you would expect, because what you are doing is clustering by their biological function, and that means that even on one gene which is anti-inflammatory, the other that is inflammatory, they may belong to the same guy. So you have to make this difference. Just to show you what happens to SARS-CoV-2, and I'm showing you this. The final thing is that we took also the Emmanuel Weiler, they have published something that they have analyzed SARS-CoV1 and SARS-CoV2, but they did RNA-seq, not a microarray, and they have a little different CALU3, and they were not very, I mean, the MOI was much lower than those used by SIMS. And here you see what happens when I compare the SIMS data, which I just showed you, with the SARS-CoV-1 for Weiler. And you see here that in the beginning at 4 o'clock and 12 o'clock and after 24 hours, what you see is that the peaks are growing, but you can see that the peaks in the SIMS are the same that are presented by in the measurement by Weiler. And because we have SIMS that goes longer, you can see what happens here when you compare 54 with 24. I mean, the transcriptogram could select which are the things that are really different. In SARS-CoV-2, we can also do that, and we can compare the differences. Okay, so we just compare the SIMS data with the SARS-CoV-1 because that's what we have done. But we compared the SARS-CoV-1 and SARS-CoV-2 for the same series, and you can see that they are there. The peaks are the same, okay? And the peaks being the same, only that the SARS-CoV-2 are more intense. So what we have seen is that for the experiments by the same laboratories with the same techniques, that in fact in CALU3, which is a lung cell line, they elicit the same peaks to grow up, only that SARS-CoV-2 are more intense. If that's good or bad, I'm not sure, okay? And sorry for okay, the conclusions is that the infection depends on the cell line. The immune response depends on the cell line. Time course of infection depends on cell culture details, because you had the different details in the quiz. Time course should extend for at least 72 hours post-infection at least. Time-matched controls are important to account for culture effects. In Transcriptogram allowed comparison of RNA-seq and microarray results from different laboratories. SARS-CoV1 and SARS-CoV2 elicit similar immune responses in CALU3 cell lines. And thank you. Sorry for having to rush again.
Thank you, Rita. I understand trying to give these mini seminars is always a challenge. Trying to squeeze research into such a short time is difficult.
Yes,
we appreciate your being willing to do that.
I really understood.
So we'll move on directly to Jim Fetter, and then we'll have time for discussion and questions afterwards. We have the extra half hour this time, which is very nice, so we have more time for open discussion.